Do you have any questions from what we did last time? If you have a question, you can ask before we start a new topic. Something I didn't understand or want clarification on. Okay, right. So it looks as pretty much we are okay with what we've done so far. On Google Classroom, I put a lot of resources that you can also have access to, so try some of them. There are some English resource and other things that you can also practice on. For the English part, do a lot of vocabulary, and reading comprehension, speed reading. So do a lot of problems online, comprehension exercises, vocabulary, and so on. That will help you. Every day, do a little bit of it so that it will be part of you. I mean, for the math and science, it's more straightforward. So it's easy to pass those sections. Okay. All right. So today we're going to do first half the science and then the second half the math. We're going to do a lot of math today. So let's start with the first topic. Now, the first topic that I want to look at is the flower. We want to look at the flower. So let's look at the flower. So I hope everybody can see what I have on the screen. If you check the Google Classroom, I also have the document over there. So you can always pull that up also and then follow along. Okay. Okay. All right, so let's go through the flower. And usually, the flower I always like to use is the hibiscus flower because that one has all the different parts that we want to talk about. Okay. So we have to note that the flower has both male and female parts, right? So you have male and female parts when you talk about the flower. Male and female parts. Okay. So every flower has that. Sometimes it may be on the same flower or it can be on different flowers, right? There's a name when they are on the same flower and there's a name when they are on different flowers, but I'm not going to mention that here. Now the male part is what we call the pistil. So the male part is what we call the stamen. The word is stamen. Stamen, that's the male part. And the female part is what we call the pistil. Female part. So the stamen and pistil. So if you look at the diagram over here, this is a pistil. There's a pistil here. That's a female part. And over here, this is what you have, pistil. And then the stamen is the male part. Made up of what we call the anthers and filaments. The anthers and filaments. So this is made up of the anthers and the filaments. And the female part, you have 1, 2, 3. So you have what you call the stigma. You have the style. You have the ovary, and then the ovule. Okay, those are the female parts, you know. So basically, that's what you have in this diagram. Okay, in the diagram here, and then the colored part is the petal. Okay, and then the calyx is the cap-like structure in which the petals sit. You know, the sepals are like the small, like they like a leaf-like structures on the calyx somewhere here. Okay, so if you draw a hibiscus flower, basically what you have there looks like this. Something like that. All right, and then you have a tube that goes in, and then the ovules are the eggs. The ovules from what they got the egg. That's ovules. And the petals are here. Okay. And then the male part will be this hanging here and that. And then that'll be the flower stalk. This how it looks like. Okay. So the anthers that you see here, the anthers, they contain the pollen, right? That's where you find the pollen. So the pollen can be found in the anthers. So let's write that here. That's where you have the pollen grains. You find them in the anthers. Okay. Right. And the filament is just this. This one is the filament. That is like the stalk holding on to the anthers. So this is the filament here. The filament. Okay. And then this will be the ovules. The ovules are the eggs. The ovules, like the eggs. Okay. In the ovary. So the ovary is this over here, like that. Okay. Then this part is the stigma. The stigma is a sticky part, of the flower. Because the stigma, okay, the female part of the female. So it's sticky. Okay. Very sticky parts. Okay. Yeah. And the style is this here. There's a style. That's a style. This long tube-like structure is a style. Okay. Yeah. So let's go through the reading here. So I have the male part of the flower is called the stamen. It consists of the anthers and filaments. The anthers contains the pollen grains, right? That's point number 1. Point number 2, the female part of the flower is called the pistil. It consists the stigma, the style, the ovary, and the ovule, right? 3, the stigma is the sticky part and it attracts the pollen during pollination, which we're going to talk about pollination shortly. Pollination, so let's talk pollination. The pollination is the transfer of pollen from the anthers to the stigma, right? So when you have the pollen coming from the anthers, so let's use red here. The pollen is here. When it is transferred from the anthers onto the stigma, that is what we call pollination. Pollination. Okay, pollination. All right. So because this is sticky, the pollen gets stuck to this, and then the pollen will begin to grow it starts to grow through the style here, and then it will go to fertilize the ovary. Okay. So that's what pollination is about. I'll talk about that again. All right. So we said that number 4, pollen is the transfer of pollen from anthers to a stigma. It may be self or cross pollination. So you can have what we call the self or cross pollination. Right. Now, when we say self pollination, it involves the same flower. Right. So like this one that I just discussed, from the anthers to the stigma, that would be self pollination, same flower. But when it involves different flowers, let's say from one flower to another plant somewhere, so that would be cross pollination, okay, or different flowers. Okay. And 5, so the pollen grows through the style into the ovary to fertilize the ovule, right? So fertilization leads to seed production and embryo formation. So seed is the fertilized ovule. This is very important to note down. Number 6, we'll come across a question using that information, right? So what you have to know that is that pollination leads to fertilization, because it's the fusion of the male part with the female part. So you have fertilization. So once you have fertilization, you have an embryo. So let's write a little note for ourselves. I think I have it somewhere in the notes too. So when you have pollination will lead to fertilization. Because it's a male gamete that will fuse with the female gamete. So once the male and female gamete fuse, then you have an embryo formed. Okay. Now the embryo, that's what you have the seed, leads to the seed formation. Because the embryo becomes the zygote and then to grow and so on. So it leads to seed formation. Okay. All right. So note, if there's no fertilization, you cannot have seed production, right? That's a very important key information. No fertilization, no seed production. Okay. Right. So please note that down somewhere. Then the petals are the colorful part of the flower, and they attract the agents of pollination. All right. So they attract the bees, the butterflies, human beings, and so on. And those are called the agents of pollination. And water, wind, they are all agents of pollination, and they can blow the pollen or carry the pollen from the anthers onto the stigma for fertilization to pollination to occur, and that will lead to fertilization. Now the ovary wall becomes the fleshy part of the fruit, right? So once you have fertilization and from the embryo and from the fruit, from the seed and the fruit, the embryo, the fleshy becomes the fleshy part. Like you take like a mango, the seed will be the fertilized ovary, and then the fleshy part is the ovary wall, the wall of the ovary, covering it. Okay, the ovary wall becomes the fleshy part. Right. So that's a quick summary of what happens in pollination. Okay. Then under this, I have the same thing here, under pollination, the summarized version. So we said that already. So it is a transfer of pollen grains from the anthers, the male part of flower, to the stigma, the female part of flower, by pollination agents such as winged animals and insects. Right. I have self-pollination. The pollen is transferred from the anthers to the stigma of the same flower or another flower of the same plant. Cross-pollination. The pollen is transferred from the anthers of one flower to the stigma on another flower on a different plant. And remember that cross-pollination gives rise to hybrid plants, like new plants. Usually they are more resistant to infection, and they thrive better in harsh conditions. Okay. So cross-pollination is very good to produce new kinds of breeds. Okay. And then finally, as I said, pollination leads to fertilization. The male gamete in the pollen grain fuses with the female gametes in the ovule. The fertilized ovule becomes a seed containing an embryo. The embryo germinates into the seedling. Right, so that's a summary of all that I've said so far. Okay. Then you have to know how the seed looks like. Sometimes they draw the seed. They draw the seed, and you should be able to label the parts. So we're going to look at the seed. I have the diagram here. So this is like the Bailey seed, So it has the features that we want to illustrate. So when you take the seed has an outer coat, a covering. It's called a seed coat. The seed coat, all right. Another name for the seed coat is the kind of with the word Tesla. Okay, seed coat. Okay. And then the bulk of the seed, the blue part here, is called the endosperm. The endosperm. The endosperm is the stored food. So this is the stored food that is needed for germination, and it's mostly starch. That is stored and to be broken down to simple sugars to provide the energy for germination. All right. So this is the bulk of the seed. And then you have within the seed, the seedling, the young, the part that will come the seedling, the first plant that will come out. And it had parts. The part called the cotyledon. The cotyledon is what becomes the leaf, the seed leaf. Becomes a leaf. So you count the leaves. Then the epicotyl, epi means above, right? And the epicotyl becomes the shoot. Okay. Then the radical is what becomes the root. Becomes the root. Okay. Another name for the radical is what we call hypocotyl. Hypocotyl. Remember, hypo means below, right? So cotyl is like the leaf, above the leaf, epicotyl below the leaf, hypo. That's where those words come in. Okay. So if the plant is growing, sometimes you see the leaf come out like this, and then you have the roots. Okay. So that's the cotyledons, the seed leaves, then the shoots, and then you have the radical, the roots, something like that. Okay. So this is a diagram that you should know very well. Sometimes they draw the diagram, they ask you, they label them A, B, C, D, or 1, 2, 3, and you to identify with which part is the endosperm or the stored food, which one is the cotyledon, which are epicotyl, radical, and so on. Any questions so far on the first part, the flower and pollination? Flower and pollination. Today we're going to go through a lot of questions in the book. Today's lesson should be pretty much a lot straightforward. Okay, so I believe so far so good for the first part. Okay. Right, great. So now let's switch our mind a little bit to physics, back to some physics. Back to physics. Okay. Now I want us to look at heat and the mode of heat transfer, how heat is transferred from one object to the other. Okay. So we all know that heat is a form of energy which is transferred between bodies which are under thermal interactions, right? Now, heat transfers occurs when temperature difference occurs between two bodies or a body with its surrounding. So remember, heat is transferred from hot to cold, right, objects. So there should be some temperature difference between the two objects for the heat to be transferred, okay, from hot to cold. And there are three modes of heat transfer. The first one is called conduction, that is this, and then we have convection, and then we have radiation. Conduction, convection, and radiation. Those are the modes of heat transfer. Okay. So what is conduction? Let's look at that now. So when you say conduction, is a heat transfer by means of molecular agitation within a material without any motion of the material as a whole. Right? So if one end of a metal rod is at a higher temperature, then energy will be transferred down the rod toward the colder end because the higher speed particles will collide with the slower ones with a net transfer of energy to the lower ones. So what we're saying is that you have an object like a metal, one that is hot. So let's say the hot one, and it's a colder one. Okay. So you have electrons in there. So these are the electrons. Okay. So the whole metal doesn't move. But what happens is that the electrons, they gain energy. You know, the temperature changes their energy. So when they change energy, they vibrate and then they hit each other. So there's vibration. So they vibrate and hit each other. And as they bombard with each other, they transfer energy from the hot one to the cold one. There's energy transfer of energy. Okay. And that's what happens. It gets here. The electrons also gain energy. It transfer the energy to this and so on. So it gets the energy transferred from one electron to the other. So the whole metal, it doesn't move. That's what we're saying here. Okay, right. So, and the second one is convection. So convection. So convection is the transfer of heat by currents within a fluid. Current within a fluid. So what we're talking about, convection. So this is for convection here. We're talking about fluid. Let's say you have a water, and you are boiling the water, put it on fire. Okay. What happens is that you have the lower part of the water warms up, and then warm water will always rise, and then its place is taken by the colder water, right? So it creates like a current. Current is created in the fluid. Okay. So the warm water rises. And its place is taken by the colder water. So that's what is happening. So like a current is created within the fluid. Okay. So the bulk of the substance moves. Okay, when we talk about convection, right? So the second part is like it may arise from temperature differences, either within the fluid or between the fluid and its boundary, which would affect the density, right? So the warm water becomes less dense, so it rises. The colder water is denser, so it sinks. So that is what creates the warming of the whole water. So after some time, the whole water becomes uniformly warmed, okay, because the densities of the cold water and warm water are different. Okay, that's convection. So there's bulk movement in convection, bulk movements, right? Then the last one is radiation. So in radiation, the heat transfer is concerned with the exchange of thermal radiation energy between two or more bodies, right? Thermal radiation. So talk using what we call radiation. Thermal radiation is defined as electromagnetic radiation in the wavelength range of 0.1 to 100 microns, right? So this is a range. If you think of visible light, the visible light is within that range, okay, and arises as a result of temperature difference between two bodies. So temperature difference between two bodies can result in radiation, when there is electromagnetic waves involved. Okay. Now, because this involves electromagnetic radiation or waves, you don't need contact between the two objects. So very important. So that's why I made this in bold. No medium need to exist between the two bodies for heat transfer to take place, right? So radiation, like the sun, if you have the sun, you don't have to have contact with the sun for the object here to feel any heat. Okay. This radiation, magnetic waves, electromagnetic waves. Okay. So you don't need any contact, right? So even as this vacuum, you can still feel the radiation. Okay. You don't need medium to exist between the two objects. Okay. So let's say the sun. Okay. So this is how heat is transferred from one object to the other: conduction, convection, and radiation. 3 modes of heat transfer. Okay. So if you take something like the flask, usually they try to reduce heat loss by reducing these 3 modes of heat transfer. For example, let me draw a flask. So if I take, let's say, this flask and draw a flask like this, if you look at the flask, inside the flask is shiny. You have a 2-walled outer layer, covering. These are shiny surface, shiny, and in between you have air in there. Okay. And then you may have the outer cover itself somewhere. And then below here, there'll be something like insulator somewhere here. Okay. So what they're trying to do in the flask is to reduce heat loss by conduction. So the conduction, when this is on the surface, the insulator will reduce heat loss from the flask onto any surface. So this will be the conduction here is reduced. By the insulator, right? Then you have air in between. This is a bad conductor of heat. right? So that's why you have air in between. So it also reduces heat loss by conduction. Then you have the shiny surface. The shiny surface here is going to reduce heat loss by radiation. So that reduces radiation. Okay, so the heat reflected into the inside part instead of going outside, because reflection inside. Okay, so this is how they applied heat loss. They try to reduce heat loss by the 3 methods that we just described in the flask. Great. Any questions so far? Let's pause for 30 seconds. Any questions?
Oh, good.
Good so far. Great. All right. So now to the more difficult part, calculations. Like if you look at the notes, sorry, in the book, there are some questions on calculation of heat energies. All right, so we're going to go through that. As I said, today we're going to have time to go through all these questions in the book. So it's a shorter lesson. Okay. Right, so quantity of heat, you have to not calculate quantity of heat, how much heat an object has. Okay. So please look at the screen for this one. So the quantity of heat, let's use Q for the quantity of heat, gained or lost by a substance is equal to the mass of the substance, which is M, multiplied by its specific heat capacity, what we call the specific heat capacity. I'll define that shortly. Multiplied by the change in temperature. Okay, the change in temperature is the final temperature minus initial temperature, right? So mathematically, that's have Q is equal to MC theta, change in temperature. Okay, so M times C times the change in temperature, final minus initial. Okay, so basically, if you're writing out, is MC delta T. Okay, mathematically, delta T means change in temperature. Okay. So what is the heat capacity? We define heat capacity as of a substance as amount of heat required to raise the temperature of that substance by temperature of 1 gram. So the keyword is 1 gram of that substance by 1 degree Celsius, okay, or by 1 Kelvin. So the amount of heat that you need to change the temperature of 1 gram of any substance by 1 degree Celsius. That is what we call the specific heat capacity. Okay. So every object has its own specific heat capacity. How much heat you need to put in to increase the temperature by 1 degree Celsius. Okay. Specific heat capacity. So that for water is usually 1 gram. It's 1 gram per 1 calorie per gram per Celsius. You know, for water, it's 1. Okay. Now the heat capacity is defined as the mass of the substance multiplied by the specific heat capacity. The mass times the specific heat capacity. That's what we call the heat capacity, right? So the first one is specific heat capacity. That one is 1 gram. This time you're talking about heat capacity. It's just the mass times the specific heat capacity. Okay, right. So with this information, we can say that the quantity of heat can therefore be defined as the heat capacity multiplied by the change in temperature, right? Change in temperature, because if you go back here, MC, this is your heat capacity, and this is your change in temperature. So that's what that means. Heat capacity combines the mass times the specific capacity. So let's look at the first example here. It says calculate the quantity of heat needed to raise the temperature of 250 grams of water from 20 to 56 degrees Celsius. Now the heat capacity of the water is given as 4.18 joules per calorie per Celsius per gram, right? This time we're talking about joules. So the joules is 4.18, okay, in terms of joules, and that's another unit for energy. So to do this, we have to use a formula. The quantity of heat is equal to the mass times the specific heat capacity times the change in temperature, right? That's what we're using here. What have you been given? You've been given the mass of the water as 250 grams, so that's the mass. The temperature, initial temperature is 20, so that's this. And then the final temperature is 56. You're heating it from 20 to 56. So the change in temperature here is a difference, which is the final temperature minus the initial temperature. Okay, that's why we have a 56 minus 20, so that is 36, right? So the change in temperature is 36 degrees Celsius. Okay. So the mass 250 times the specific heat capacity, which is 4.18, then times the temperature change, 36. So if you multiply this out, then you get 37620 joules. So 37,620 joules, right? So joules is a smaller unit. So we're going to change the bigger kilojoules. If you change to kilojoules, depending on the answer that you're given or the unit that you're asked to use, then you may have to convert. So remember that 1 kilojoule is equal to 1,000 joules. That's a conversion if you have to convert. Okay. So if you have to change these joules to kilojoules, you have to divide by 1,000, and then that gives us approximately 38 kilojoules. But that would be 37.620 if you round up to the nearest whole number in 38. Okay. So that's how you work out this problem. Remember the formula: mass times specific heat capacity times the change in temperature. That's the key. Any question on that one? Okay. All right. So let's test ourselves on this question. I have questions for the day. So try B. Try B. Take like 2 minutes to try it. Right. Did anybody get any particular answer? All right, so if you go down here, I have the answers down here. So you've been given the heat capacity. Sorry, you want to calculate the heat capacity. Given that 204.75 joules of energy raises the temperature of 15 grams of copper from 25 to 60 degrees. All right, so you have your final temperature, you have your initial temperature, you have the energy, and then you have the mass. All right, so if you go back, this will be the energy is equal to, that's your Q, is equal to 15 times specific heat capacity times the change in temperature, you know. So if you work this out, you should come out to C equal to this, okay. You find in C, which is missing, okay. Were you all able to get that? Were you able to get that, everybody? So if you didn't get that, you can let us know. Okay, all right. So you should be able to practice on the other ones later, you know. Make sure you get these answers. All right. Great. So the other aspect of heat that I want to look at is what we call the change of state. Change of state. Okay. So we know we have the three states of matter: liquid, gas, and solids. Liquid, gas, and solids. Right. Remember that gases have no fixed shape and no fixed size or volume properties. Gases, no fixed shape or volume. Liquids, they have fixed volume, but no fixed shape. And then solids, they have fixed volume and shape. Fixed volume and shape. Okay. Properties. Right. So we can change one form to the other, right? Change one form to the other. So remember, when changing from gas to liquid, that's condensation. When you're changing from liquid to gas, that's your boiling or evaporation. If you're changing from gas to solid, it's called resublimation. And if you're changing from solid to gas, that's sublimation. Okay, they like this word too, sublimation, very important. Changing from liquid to solid, that is freezing, and from solid to liquid, that's melting. Right, those are the change of states. Okay. Example of sublimation, you have to know example of sublimation. Example of this would be if you have what we call a dry carbon dioxide. Dry carbon dioxide, if you have it and leave it for some time, it will change from the solid into the gas without becoming liquid. Okay, that was sublimation. Okay. Another example would be if you think of the mothball, or some people call it camphor. It's white. You know, people use it to drive away like roaches and other insects. You know, put in the drawer in the corner, you know, drives them away. Yeah, it's white. So if you put it down, it doesn't become liquid. It just changes to gas, you know. So that is an example of sublimation. Okay. Another example would be like the air refresher, the solid one. If you take that one, it doesn't become liquid. It's just solid, then just turns into gas. And you know, those are all examples of sublimation. Right. So think about some examples like these. Okay. Right. All right. So the change of state, what is the energy that is involved? Right. So let's go to the diagram below. Know this diagram very well. Change of state diagram. Change of state diagram. Okay. So let's begin by looking at water that is at negative 20 degrees, right? So let's say I have water at negative 20 degrees. So that ice is going to be ice at that temperature, negative 20 degrees Celsius. Okay. Now, when I begin heating up this water or warming up the water, what's going to happen? This temperature is going to increase. As you can see, it's increasing until it gets to a temperature of 0 degrees over here, Celsius, right? So it's going to increase till it gets to 0 degrees Celsius. So to still be ice as you warm it up. Then something's going to happen. As I put in more heat, you are putting more heat. As you put in more heat, it begins to melt. So the ice begins to now melt. But the temperature doesn't change. It stays at 0 for some time. So it's 0 for some time, even though you're putting heat, it's staying at 0 for some time, get like a plateau. So it becomes liquid, but then the temperature is still 0 degrees. Okay. And the reason will be why. So here it's melting here. So what is happening here? Now, what is happening here is the fact that if the heat that you put in is used to break the bonds between the molecules, right? So you're breaking the ice is solid, so there's strong attraction between the molecules. That's what to maintain the fixed shape. So the energy goes to break the bond between the molecules, or the molecules, and then freeze them so that now they can move a little bit further from each other to become the liquid. So the liquid has no fixed shape, right? That energy that you put in is what we call the latent heat. So you don't see, latents are hidden. So we call this the latent heat of fusion. Latent heat of fusion. Okay, that's the heat that I put in for this to to melt. Okay, so melting was occurring here. Latency of fusion, right? So apparent heat that you don't see. It's doing some work you don't see. So the temperature doesn't go up. It stays at 0 for some time, and that's what this is. Okay. So it's a change of state here. Change of state is occurring here, and that is a latent heat. Of fusion. That's what we call it here. Okay. Then as you put in more heat, the temperature will now shoot up. So the temperature increases till it gets to 100 degrees Celsius, 100 right here. So now the liquid temperature will increase to 100. Degrees Celsius. Okay. Putting more heat, what's going to happen? The temperature is going to increase again. As it, sorry, the temperature will not increase at this point. It's going to stay at 100 again, but this time it's going to become vapor or gas. Sorry, it's going to be vapor. So it goes to a gas phase or vapor. Okay. But you're going to realize that the temperature doesn't increase at that point. It takes some time. It stays at 100 degrees Celsius for some time, even though you put in more heat. What is happening here? Now, again, at this point, the heat is used, the heat energy is used to break the bonds between the liquid molecules, okay, to free them into the gas phase so they can move a little bit further away, so it becomes gas. It's breaking the bonds. So that's why the temperature doesn't increase for some time, right? And that's here, and that's what we call the latent heat. Latent heat of vaporization. Okay, that's what's occurring here. So this here is a latent heat of vaporization. All right. So all the energy is used to break the bonds. So the temperature doesn't increase for some time, stays 100 for some time. So it's a change of state here, a change of state from one form to another, right? And then once it becomes a vapor, the temperature will now increase above 100, can go above 100. Okay, then it can be a gas or vapor with temperature more than 100. Okay, right. So this is what is happening. The flat is change of state occurs where they have the flat lines. Okay, so note this diagram carefully. We'll see this again in the book as we answer questions. Okay. Any questions on change of state? Okay. So the diagram helps to explain everything here. So liquid and solid, yeah, equilibrium, latent heat of fusion. If you come to latent heat of vaporization, the liquid and vapor, they are in equilibrium with each other at this particular point, change of states. Okay. Right. Now this diagram can come in another form where it is sideways. It can come in a sideways form like this one. Let me see if I have this in the sideways form. All right, this is the same diagram, but in we have change of the phase change written as part of this. Yeah. So latent heat of sublimation, if it's changing from solid to gas, then again that can also occur there. You have latent heat of fusion of vaporization, and then you have latent heat of fusion. The summary of what I've said so far. Okay. Hidden energy. So the temperature has increase in those phases. Okay. As I said, this diagram can come in the sideways form, like the other way around. But if you understand it, you should answer questions on this easily. You could have it like this. So let's say that you have the energy here, and then you can have the temperature on this side. Okay. So the same thing, let's say that if this is negative 20, you have to heat it up, energy goes in, then it stays at 0 degrees for some time. So you put in energy, it stays as the same 0 for some time. Then putting more energy, the temperature can increase. So this will be 200 degrees Celsius. And then once it gets 100, it stays at 100 for some time. Change of state. And then it will increase again. And then increases again like that. So it's the same diagram, but sideways. Yeah. So change of state occurs here. This way you have the change of state. And then change of state occurs here. Same temperature for some time. Okay. So this will be the latent heat of fusion. And then this will be the latent heat of vaporization. Okay. So once you understand the diagram, it doesn't matter how they present it, you should be able to answer questions on it. Okay. Right. Great. So that is the phase, what do you call it? Energy change diagram. Okay. Right. Yeah. There's another type of diagram that you also have to know, and that is what we call the phase diagram. The phase diagram. So this is the phase diagram. Phase diagram. Okay. Now the phase diagram is easy to answer questions using the phase diagram, the question that they'll ask you. So let's look at what this represents. Now the phase diagram shows in the lab, you measure the pressure against the temperature, during the change of state for a particular substance like water or any other liquid. Okay. When this is performed at the lab, you come up to graphs like this, 3 lines that intersect at a point. Now this line here represents the line between the solids and then the liquid part of water. And then this line here represents the line between solid and water vapor. And then the line here, CB, represents the boundary between the liquid, which is water, and vapor, right? So 3 regions bounded that the lines separate. Okay. So what does this line mean? CB. CB means that if I have an object here, let me draw again on the side here. Let's say I have something like this. So this is solid. This is liquid region. And this is this vapor region. Okay. Now the question that they can ask you, usually they'll draw, let's say A to B, and then probably C to D, and vice versa. Maybe E to F. something like this. Then they can ask you which one represents sublimation. Sublimation. Okay. Now, if you go back to this line here, I said I have here melting curve. This is a melting curve. Right. This line is a melting curve. So that's a melting curve. This line here represents the vapor pressure curve, and then this line represents sublimation curve, right? And the point where they intersect is called a triple point, the triple point of that substance. So this is the triple point here. You know, for water, the triple point is 273.13 Kelvin. Okay, that's where they had that temperature Kelvin from, 273 Kelvin. The triple point means that all the phases, liquid, solid, and the gas, they're in equilibrium at that point. Okay, they all exist, they coexist at that point. Okay, that's what that means. Yeah. So if they ask you which one represents, let's say, sublimation. Sublimation. So sublimation, that means from solid to gas, right? Solid to gas. So that would be C going to D. That would be sublimation. C to D. That's sublimation because change from solid to gas. Which one represents evaporation? Evaporation. So for evaporation, we're changing from liquid to gas. So there's a liquid phase is here. So liquid and then to gas, to vapor. So it's E to F. E is the liquid phase going to vapor, which is the F, you know, instead of E to F. Which one represents, let's say, freezing? Freezing. So freezing, we know we are moving from liquid to solid, right? Liquid to solid. So freezing will be liquid, which is the B, to solid, going to A. So B to A, and so on. Okay. So that's how you can use this graph to answer questions like this. Okay. Which one represents melting? Melting is from solid to liquid. So the solid phase, which one is the solid? C to liquid. Sorry. So it's A to B. So it's melting is from A going to B, and so on. Okay. So that's how you use this diagram to answer questions. Okay. So you don't need to know too much about this, just the regions, which one. The diagram will tell you solid, liquid, and gas. They'll show you the solid, liquid, and the gas, and then you should be able to determine what each one represents. Okay. Great. So let's read this one. One says what? In thermodynamics, the triple point of a substance is the temperature and pressure at which the three phases, gas, liquid, and solid, of that substance coexist in thermodynamic equilibrium. Okay, they're all existing at that point, triple points. Okay. And then two, what do you mean by a supercritical fluid? So SCF. A supercritical fluid is any substance at a temperature and pressure above its critical point. Above its critical point. So if I take this, for example, the temperature here, let's go back here. This is a critical point. So if I take a point here, it's above the critical point, the temperature above the critical point. Okay, so this liquid will be in the supercritical, will be a supercritical fluid, and so on. Okay. So in other words, a supercritical fluid is any substance at a temperature and pressure above its critical point, where distinct liquid and gas phases do not exist. You cannot distinguish between the liquid and gas phases. Yeah. Again, it can effuse through solids like a gas and dissolve materials like a liquid, right? So it's neither like a liquid, then like a gas. You can't distinguish the two, so they can easily go through solids as if it's like a gas, or dissolve materials as if it's like a liquid. But for the test, mainly know how to the regions, like from A to B, B to what does it represent. Great. All right, any questions on using this diagram? We'll see that shortly in questions. Any questions so far? Okay, all right, great. All right, so let's look at another aspect. Now let's come back to some biology. Okay, so that'll be the last, I think the last topic for today's lesson on science, so that we can answer questions. All right, genetics. So that should be pretty much straightforward for us. So we want to talk about inheritance. Okay. So we know that chromosomes carry the hereditary information, which is the genes, and we know that they are arranged. They are determined by what we call the nucleotides in the DNA molecule. Okay. So the genes are the arrangement of nucleotides in the DNA molecule. Okay. We discussed the DNA molecule, the difference between DNA and RNA. I believe we did that the first day of class. So we'll skip that part, But just for a quick review, we know that nucleotide, when I say nucleotide, we said it's made up of the nitrogen base and the sugar and the phosphates. That's what forms the nucleotide, if you remember that. Okay. And then we spoke about the nitrogen bases, guanine, adenine, cytosine, thymine, okay, in DNA, and then your RNA, you have all of that except thymine. Uracil replaces the thymine, which we discussed, right? Okay. So just a quick review, remember to look at that again. Okay. Right. So the chromosomes and the genes occur in pairs. They occur in pairs. So that's what we call the homologous chromosomes. So if you look at the DNA, you have on the chromosomes, they occur in pairs, like a double helix, something like that. So you have a gene here, there's a corresponding gene at the same locus. Locus is the position of a gene, right? So those will be the homologous chromosomes, two chromosomes that look alike. Okay. And see, new combinations of genes occur in sexual reproduction, right? Fertilization from two parents. So when there's fertilization, there's mixing of the genes, and that's what we're going to look at. What are the offspring that will come out of that? Some terms to know. You have to know what a gene is. The gene is a unit of heredity, a section of DNA sequence encoding a single protein, right? So this would be like a gene. Let's take on the height. So the height, the gene, let's say uppercase T for tallness, and then we can have maybe this for tall, and then let's say T, lowercase t. Let's say that that's a gene for short. Okay. Then they will carry the same position on the chromosome like this. So they'll be the homologous chromosomes. Okay. Alleles. So when I say alleles, we're talking about two genes that occupy the same position on the homologous chromosomes and cover the same trait. So example, if you talk about height, so for height, as I've just mentioned, the homologous chromosomes will be T, uppercase T and lowercase t. That would be the homologous chromosomes. Okay, homologous chromosomes. We use the uppercase for the gene that we refer to as the dominant gene, and the lowercase for the gene that we refer to as the recessive gene. So this will be the dominant gene, uppercase, dominant gene. And then the lowercase will be the what we call the recessive. Dominant and recessive genes. Okay. Now the dominant gene is a gene which will always show itself in the person. All right, it always shows itself. So it's dominant. So the trait that appears in the heterozygous condition. Okay, I'll explain what heterozygous means. Okay. And then recessive gene is a gene which is suppressed, right? It's suppressed. It will only show itself if it is the only gene present. The only gene present to show itself. Okay. Otherwise, it will be suppressed. Okay. So let's go to 3. It's a locus. It's a fixed location on a strand of DNA where a gene or allele is located. So this position will be the locus. The locus where you find a gene, the position. It's a locus. Okay. Then homozygous. Homozygous means that they have the same genes at the locus, right? Same gene from one from each parent for particular characteristics. So if I take something like height, if the person have 2 dominant genes, T and T, then the person is homozygous. Or if the person has lowercase t and T, then the person is homozygous. Same genes as the locus. Right. Heterozygotes, 2 different genes. So you can have uppercase and lowercase. Then this person will be heterozygous individual. 2 different characteristics. Okay. So the phenotype is the outward expression of the gene, what you see on the outside, how the person look on the outside. So if you take something like uppercase, if the person has homozygotes for the gene for tallness, then this person is going to be tall on the outside. If the person has uppercase T and lowercase t, because we said the tallness is dominant, it means that it's going to suppress the lowercase. So this person outwardly, even though it's heterozygotes, 2 different genes, this person on the outside is going to be tall because tallness is more powerful, dominant. Okay, so that's what is on the outside. If the person has 2 lowercase t's, then this person is going to be short. Okay, so the recessive gene will only show itself if it is the only gene present, like, all right, only gene present. It's suppressed in the heterozygote state. So that is a phenotype, outward expression. Then the genotype is the genetic makeup, what the person is made up of. So the person, let's say, is made up of either TT, homozygote, or TT, or the person made up of this, the genetic makeup, homozygous or heterozygous. Okay. And then we also talk about phenotypic ratio and then genotypic ratio very soon. Okay. The ratio of what you see on the outside, or the ratio of what the genes are made up of. Okay. So these are terms that you should know to answer questions on the test. Okay. So let's see how we can apply these concepts. So that takes us to what we call the Mendelian inheritance, or Mendel's laws of genetics. Okay. So Mendel was a priest, and also he did a lot of scientific work, scientist as well. He studied a lot of genetic processes. And he came out with some laws. And the first one is Mendel's law of dominance, which says that when an organism has two different alleles for a trait, one allele dominates, as we just said. So uppercase and lowercase, the dominant with the tallness, which is uppercase. We use uppercase for dominance. Then it had Mendel's law of segregation. Mendel's law of segregation. It says during gamete formation by a diploid organism. Diploid organism means it has two genes, right? Two pairs of genes. Like human beings, we are diploid. In the cells are diploid, whilst in the body cells, while in the sex cells, you have haploid. Haploid means just half the number of chromosomes. Haploid, all right? So the egg will have like half, the sperm will have like half. It'll be haploid. If they combine, they form a diploid. So it says during gamete formation by a diploid organism, the pair of alleles for a particular trait separate or segregate during the formation of the gametes, right? So what he's saying is that, let's say that if this individual has uppercase, let's say this, and then let's say that's a man, and let's say female is uppercase T. During gamete formation, the gene separates into different gametes. So one will go into one gamete to have that. So let's say one sperm has this, and another sperm will have that. Then if you take the egg, one egg can have this uppercase, and another will have uppercase. They separate like this. Okay, that's the law of segregation. Okay. They separate or segregate during the gamete formation. So this will be the gametes. The gametes. Okay. Yeah. So that's the segregation. Okay. Then his third law is the Mendel's law of independent assortment. Independent assortment. So assortment means that there's random combination of these gametes, random combination of these gametes. So it says members of a gene pair separate from one another independent of the members of the other gene pairs. These separations occur in the formation of gametes during meiosis. So it's randomly processed. All right, so let's look at an example, and then they combine randomly. Okay, random combination. This can combine with that, this can combine with that, and so on. Random combination, our assortment. So let's look at an example. So let's say that you have a female who is homozygous. So female let's say homozygous for tallness, marries a male who is heterozygous for tallness. The question is, what will be the genotypic and phenotypic ratios we want to find the phenotypic and genotypic ratios. What would this be? Okay. So again, you just have to understand the keywords that are used here. So if the person is homozygous female, so it means that this female, the genes that the person will have will be, so let's use this symbol is for female. Okay. So it means the female is going to be homozygous for tallness, so uppercase T, right? And then the male, this is a symbol for male. The male will be heterozygous for tallness, so the male, the genes will be uppercase and lowercase. Okay, that's what the male will be. Okay. Now, to answer this question, you can use what we call the Punnett Square. So we're going to draw the Punnett Square for this. Use the Punnett Square. Okay. So the Punnett Square, you draw a diagram like this. And then you're going to put in the female on one side and then the male gamete on the other side. Okay. Now the female is uppercase T. So the two genes, you separate them based on the law of random segregation. So it means that they can go into different gametes. So let's say this one gamete, so T and another T will go here. Okay. Then for the male, you have uppercase and lowercase. So if they separate, one will go into one gamete, which is this box, and then the other one separates into another gamete like this. Okay. Then there's random assortment or combination. So the members will combine, the genes will combine. So we can have this combined with that. So you have uppercase, this will combine with this, uppercase and lowercase. Then this can combine with that, uppercase, then lowercase and uppercase, T. So these are all the possible combinations that you can get out of this, all the possible combinations, right? So the question is, what is the genotypic ratio, genotype? So you're going to get genotypic ratio. Okay. So you're going to look at the type of genes. If you look of the, so these are called the offsprings, right? Offsprings. So if you look at the offsprings or the first generation, usually we use the word F1. Okay. Here we have two genes that are TT, uppercase T. So 2 out of a total of 4 possibilities, 2 out of 4. So you have 2 out of 4 possibilities of uppercase T's. So that's homozygous individual for tallness. Then the other one, 2 out of 4 possibilities, 2 out of 4 of uppercase and lowercase T, this heterozygote condition, right? In other words, you have 50% TT and then 50% TT, 50-50 of homozygous tallness and heterozygous tallness. Okay, that would be the ratio. 50%, 50% homozygous, and then you have this heterozygous. Okay. In other words, it's like 1-to-1. If you look at this ratio is the same as saying that you have 2-to-2, which is 1-to-1, 2-to-2, or 1-to-1 ratio, which is the same as 50-50. Depends on what you're asked, whether it's percentage or fractions and so on. So that's how you use the Punnett Square to answer a question like this. Okay. So the other question will be, what is the phenotypic ratio, phenotype? What is a phenotype, phenotypic ratio? Phenotype. Okay, so when it comes to a phenotypic ratio, you look at what they will show on the outside, right? So let's go back to our list here. If you go to the Punnett Square, now you have TT1, this will be tall, T uppercase TT, this will be tall, and then this will be tall, and then this will be tall. So it means that they're all going to be tall because the uppercase is dominant, okay, tallness is dominant. So they're all going to tall. So here we have 100% tall. All of them are tall, all tall. So that would be the ratio here. They're all tall. All tall. What they show on the outside. Okay. Now let's see if let's change this question and put in something else. Okay. So let's say that now you have a female. This time, instead of a person being heterozygous, let's say that this person homozygous. Let's say this person is heterozygous. So I'll change this to heterozygous. So let's say they have heterozygous individual. For tallness, and then marries heterozygous for male. Okay, so let's see what's going to happen here. So we use the Punnett Square again. All right. So the female heterozygous, so it's uppercase and then lowercase t, and then the male will be this. So the Punnett Square will look like this. So if you had a female here, let's put male there. So uppercase and lowercase, uppercase, lowercase here. If you combine this uppercase TT, then this will be uppercase and lowercase, to lower, and this is uppercase and lowercase, and this will be lowercase. Okay. All right, so this is what we have for Punnett Square. So the genotype or genotypic ratio will have 1 out of 4 homozygous for tallness, and then the other one will be 2 out of 4 heterozygous, and then the other one is 1 out of 4 homozygous for shortness, right? So if you look at ratios 1 to 1, It's like 1 to 2 to 1 type of ratio, 1 out of 2, 1 ratio. Okay. Or in terms of percentage, this will be 25% homozygous TT, and here you have 50% heterozygous for the height, and then you have 25% homozygous for shortness. Okay. So this will be the genotypic ratio. Okay. Now, what would you see on the outside? The phenotype. Phenotypic ratio. Okay. What am I going to see on the outside? So on the outside, when I go here, this person is going to be tall, because tallness is dominant. This is another tall person, because uppercase is dominant. And this is another tall person, uppercase dominant. So 3 tall out of 4 possibilities. So 3 out of 4 will be tall. 2, 1, this person will be short. 1 out of 4 possibilities short. Okay, because that's a short one here. Short. In other words, we have 75% of them will be tall and 25% will be short. So that's how you use the Punnett Square to answer information like this on the test. Now, sometimes on the test, they'll ask the question backwards. The backwards will be, they'll give you the ratio. Let's say that they tell that the offspring, you have 75% tall and 25% short. Then you have to work backwards to determine the parents. You know, what would be the genes or the parents that can give you this combination? So it's like working backwards. So in that case, you can work backwards using the answers that have been given to you. You do the crossing and see if you can get the ratio. Okay, so that'll be the backwards approach to answer this question. I believe we're going to see one question like that shortly. Yeah. So that should be enough for the Punnett Square, how to use the Punnett Square to generate the different possibilities. So what we discussed so far is the complete dominance, what we call the complete dominance, okay, where the gene is completely dominant over the other. There are some questions, some situations where you have what we call incomplete dominance. So I'm going to look at that quickly. I think on the test, mostly they'll ask more of what I've just discussed. Yeah. But special cases. Now, incomplete dominance. Okay, let's read that. So in some allele combinations, dominance does not exist. Instead, the 2 characteristics are equally expressed. For instance, if you take the snapdragon flowers, they display incomplete dominance in their color. There are 2 alleles for flower color, 1 for white. So they have the white one, and they have 1 for red, white, and then the 1 for red. So when the 2 alleles for white are present, the plant displays white flowers. When the 2 alleles for red are present, the plant has red flowers. But when 1 allele for red is present with 1 allele for white, the color of the snapdragon is pink, right? It's pink. So here you have what we call the incomplete dominance. Incomplete dominance. Okay. So basically, what we're saying is, let's say that you have the gene for white. Let's say white is W for white, and then red, R for red. Okay. So if the plant has WW, then it will be white. If it has RR, homozygous, then it will be red. But you have WR, since they're all equally dominant, they're co-dominant, neither of them is more powerful than the other. There's like a mixture of the two colors. So it appears the color becomes pink in the flower thing. Okay. So when I say incomplete dominance, it doesn't take on the either of those characteristics if both genes are present. Okay. It's like a mixture of the two, the shade of color in between the two. Yeah. Incomplete dominance. If it's completely dominant, then the other one will show itself, than the other. Okay. Then the other special case is called the multiple alleles. Multiple alleles. Okay. So it says in certain cases, more than two alleles exist for a particular characteristics. Even though an individual has only two alleles, additional alleles may be present in a population. This condition is known as multiple alleles. So there may be more than one gene controlling some characteristic in the individual. An example is a blood group. You take a blood type. There are more than one type of gene that controls those genes. Okay. So it said in humans, blood groups are determined by a single gene with three possible alleles. We have A, B, and O. The red blood cells contain two antigens, the A and B. The presence or absence of these genes result in four blood types. So you have the A. So the person is A, or it can be B, or it can be AB, or the person can be O. If a person's blood cells have antigen A, the blood type is A. If the person's red blood cells have antigen B, the blood type is B. If the red blood cells have both antigen A and B, the blood type is AB. If the red blood cells have neither antigen A or B, then the blood type is O. Right? So this is an example of multiple alleles. Many genes controlling different, the same characteristics in different individuals. The alleles of type A and type B are co-dominant, right? That's why you can have AB and equally powerful. That is, both alleles are expressed. However, the allele for type O is recessive to type A and B, so they don't have the antigen. They're recessive gene. So because a person has only two of the three alleles, the blood type varies depending on which two alleles are present. For instance, if a person has A allele and a B allele, the blood type will be AB. If a person has two A alleles or A and one O allele, the blood type is A. If the person has both B alleles or B and O allele, the blood type is B. If the person have two O alleles, the blood type is O, and so on. Right. So this is an example of multiple alleles. To simplify this in terms of notation, we use, let's say, AI. So this would be like if the person is IA, if the gene is IA, if you see this, then this person is A. That's what we're trying to say. So this person has a group A. Okay. If a person can have A, and let's say the gene with AB, then because they're co-dominant, they have AB, and so on, at both genes. If a person has B and then B on the chromosome, then the person is group B, and so on. If the person has neither of these, so let's say nothing on it, nothing on the other one, then it's O. Okay. So these are multiple alleles controlling. Now, the same thing here, if the person has, if we come here, if the person has this gene, A, and the other one has nothing on it, then the person will be A. The same if we come here, the person has B and the other one O, has nothing on it because this is recessive. The gene IO is recessive, so the person will be B, and so on. And that's why I say multiple alleles giving you the same characteristics. This and that give you A. This and that gives you B, and so on. Multiple. And then the other special case that we should also look at is known as the sex-linked inheritance. Sex-linked inheritance. And that one has to do with the X and Y chromosomes. X and Y chromosomes. So there are some genes that are on the X chromosomes. The Y chromosome hardly carries any genes, you know. So let's read this statement here first. It says, so among the 23 pairs of chromosomes in human cells, one pair is the sex chromosome. The remaining 22 pairs of chromosomes are referred to as the autosomes. So we have the autosomes and the sex chromosomes, right? 22 pairs and one pair, right? X and Y chromosome. So they determine the sex of the individual. The sex chromosomes determine the sex of humans. There are two types of sex chromosomes, the X chromosome and the Y chromosome. Females have two X chromosomes. Males have one X and one Y chromosome. And then, so typically the female chromosome pattern is, sorry, is designated as XX, while the male is designated as XY, XX and XY. So note we inherit one from each parent, one from each parent. So it says in humans, the Y chromosome is much shorter than the X chromosome. Because of this shortened size, the number of sex-linked conditions occur. Because you have something on the X chromosome, nothing to balance it in the Y chromosome because it's short. So you can have a lot of abnormal conditions. So when a gene occurs on the X chromosome, the other gene of the pair probably occurs on the other chromosome. Therefore, the female usually has two genes for the characteristics, you know. Because a woman has XX, usually you have another gene to counteract the other one. In contrast, when a gene occurs on the X chromosome in the male, there's usually no other gene present on the short Y chromosome. Therefore, in the male, whatever gene is present on the X chromosome will be expressed. An example of this is color blindness. If you take color blindness, there's a gene for color blindness is found on the X chromosome. A woman is rarely colorblind because she usually has a dominant gene for normal vision on one of her X chromosomes. However, a male has the shortened Y chromosome. Therefore, he has no gene to offset a gene for color blindness on the X chromosome. As a result, the gene for color blindness expresses itself in the male. Okay. So basically what we're saying is that, let me write that in notation form. If let's say they have a chromosome X, and let's use C for color blindness. Let's say uppercase C, dominant gene on the chromosome. So let's say C. If a woman has X, then this person is normal female. All right. If the person has X uppercase and then X lowercase c, this female is going to be normal, but then it's known as a carrier. It's carrying the abnormal gene. So we call that person a carrier female. So she's normal. We don't have colorblind. We don't have the color blindness, but just carrying the gene, the abnormal gene. You can transfer it to offsprings. So the male is uppercase, has a the uppercase gene, which is not colorblind, and then has Y. This will be normal, male. and the person has a lowercase, which is a colorblind condition gene, because there's nothing to balance on the Y, this person will be affected. So the male will be affected. But nothing to balance it. Okay. The same thing happens when you take the another example, the hemophilia disease, where they have abnormal clotting conditions. Okay, I think it runs through the royal family. In hemophilia, the blood does not clot normally because an important blood clotting protein is missing. The gene for hemophilia occurs on X chromosome. As females have X chromosomes, one X chromosome usually has the gene for the normal blood clotting. Therefore, the female may be a carrier of hemophilia, but normally does not express hemophilia. Males have no offsetting gene on the Y chromosome, so the gene for hemophilia expresses itself in the male. This is why most cases of hemophilia occurs in males. Okay, got nothing to balance it. So it's similar to this, what I've just discussed here, very similar. So if we take what I just described, if we come here, the same thing. If the female has, let's use H for hemophilia. So have uppercase H, uppercase H on both genes, on both chromosomes. Then the person is normal female, no hemophilia. Uppercase and then lowercase, and this is a carrier because this is normal, so the person will not have the disease, just carries the gene. Normal male, that means the male has a normal gene, then no hemophilia. Affected male, once the person has the affected gene, abnormal gene, there's nothing on the Y to balance it, so automatically this person has the disease. So that's why it's common in the males, no normal gene to offset the abnormal one. Okay. So those are sex-linked inheritance. So you can have a question where they ask you something like, a normal female maybe marries affected male or marries hemophiliac male. So let's say normal female, let's put it there. Normal female marries hemophiliac male. What would be the genotypic and phenotypic ratios? What would be the phenotypic and genotypic ratios? All right, so we have to determine the, so once they mention hemophilia, you have to know that it's a sex-linked inheritance, or sometimes they'll tell you in a question, right? So you're going to use X and Y in this case because sex-linked. So the normal female, since it's normal, we expect the normal female to be X uppercase H and X uppercase H. That would be the female. And then the male is affected male. So it means he has X lowercase h and then a Y chromosome. So that's a male. And that's a female gene. Then you can use a Punnett square to come out with the possibilities. So let's draw the Punnett square here. Okay, so for the female, we have X, H, X, H, and for the male, we have X, lowercase h, and then a Y. Okay. So when they fertilize, combination will be X, H, X, H. This will be X, uppercase H, and X, lowercase. And then this here will be X, H, and Y. And then this will be X, H, and Y. So these are all the possible combinations. Okay. Now, to answer this question, usually you try to split this up into male and female when you're analyzing, right? Male and female, so the different genders. So here, we're going to call them males and females. So among the females, what's going to happen among the females? These are the females. Okay. So if you look at this, both females, the females are all carriers. You have carrier females, right? So the phenotype or the phenotypic ratios, you have all carriers, all female are carriers, or the female, 100%. All right, now 100% carriers, carrier females. Okay, that's what that means. And then when you look at the males, XY are the males. So XY are the males. So for the males, if you look at that, they're all normal. They have carried the gene for, they don't carry the gene for hemophilia, so they are normal. So 100% healthy males or normal males. 100% normal males. Okay, so that's what we have for this scenario. 100%. Yeah. Okay. Now let's flip the information and then try another one where we have, let's say, normal. Let's go back here. And then let's have a normal. Okay, let's say carrier female rather. Let's say the female is a carrier. So X, H, and XH, carrier female. And let's say that the male is normal. So let's say normal male, X, H, and Y. Okay. What is going to happen if they produce offspring? So let's see. So they produce offspring. So we have XH, 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 and Y. So this will be XH, XH. This will be XH, lowercase. This will be XH, and then Y. And this will be XH and Y, right? So what's happening here? Phenotype. Let's look at the phenotypes here. So if you look at the females, so that would be the females here. XX are the females. So among the females, we notice that we have half of them will be normal and half of them will be carriers. So 50% female normal, and then 50% female will be carriers amongst the females. Okay. Now, what about the males? So that is one. What are the females? The males. Among the males, you have 50% will be normal. 50% will be normal males. So these are the males, normal, and then you have 50%. Half of them will be affected. So 50% will be affected males. Affected males. Right. So this is what you have when you have a question like this. Okay. Or anybody has any questions on this? Any questions on this?
I don't have any questions.
All right. So everybody's fine so far? Any person? Okay. So this is the last topic for the science for today. Okay. Right. So with this information, let's go through some questions in the book, and then we'll take 5 minutes break and come back to do the math part. All right. So let's go back to the textbook to practice some questions. Let's look at page 221, 221. Number 17 and 18. So think through the problem and then you can tell me the answer.
Excuse me, what page?
Page 221.
Thank you.
Yeah. Number 17 and 18. All right, any ideas on this one based on what we studied? And they have 50 calories here. I'm trying to draw the diagram.
I would say A.
Okay, say A. Okay, let's see. We said 540. Okay, so it says the number of calories needed to change the liquid to steam. So you are changing liquid to steam at its boiling point. Boiling temperature is, okay, 540, 590, 640, 750. Right, so you're correct. So The answer is 540 calories, correct. But you're looking at the change of state. Change of state. This 100 degrees. So there's something occurring here. It's changing from liquid to gas because it stays 100 for some time. So latent heat. That's the latent heat of fusion, of evaporation. Correct. Of vaporization. Good. And then number 18. It says the temperature were raised to 300 degrees Celsius. So now you're going 300 degrees Celsius, way above 100, somewhere here. It says asking you, the substance would A, change back to liquid, B, remain a gas, C, condense, D, break into a constituent element. Change back to liquid, remain a gas, condense, and break into its constituent elements.
Remain a gas?
Yeah, it's going to remain a gas, correct, because this is a gas, just change of state here, so now it's a gas here. So if you take the temperature somewhere here, it's going to remain a gas, correct. So it will remain a gas. Great. All right, let's look at number 19. In a field of pure white asters, a grower noticed a single blue aster. How would the grower test whether the change affected the gamete-producing cells of the plants? A, self-pollinate the flower and plant the resulting seeds. B, examine the blue petals under electron microscope. C, perform a chemical analysis to find the nature of the blue pigment. And D, perform vegetative propagation on a cutting of the plant.
A, but I'm not sure.
A, you are right. A. So you have to self-pollinate the flower and then plant the resulting seeds. If you pollinate, there is going to be fertilization. So the genes are going to mix up, right? So you don't fertilize. And the fertilization results in embryo formation that will lead to the seed. You plant the seed. And then if it grows, they determine whether there's any, whether the change affected the gametes. So self-pollinate and then plant the resulting seeds, correct. All right, number 20. A human egg will develop into female if it is fertilized by a sperm containing A, X chromosome, B, Y chromosome, C, X and Y chromosome, and D, an XXY chromosome. The egg will develop into a female if it is fertilized by a sperm containing which of these?
A, only X.
Yeah, X chromosome, because the egg contains X chromosome, so it needs to fertilize by X chromosome from the male to make it a female, correct? So X chromosome. Yeah. Right, good. So far, so good. Let's look at page 227. 227, number 51 first. Number 51. So look at page 227, number 51. Okay. So based on the diagram looks like it's a phase diagram. Pressure here, temperature there. Okay. So which answer are you choosing here?
C.
Okay, so this is 120 here. This is 100 degrees Celsius. This is 0 degrees Celsius here. Okay, so what's C? Okay, the boiling, if the pressure on water is increased from 1 atmosphere, so this is 2 atmospheres here, 1 atmosphere. It's increased from 1 atmosphere to 2 atmospheres. How, if at all, is the boiling point affected? Then A, the boiling point decreases by 20 degrees Celsius. B, the boiling point is not affected. C, the boiling point increases by 20 degrees Celsius. And D, the boiling point increases by 120 degrees Celsius. And you said C. All right, so let's see. C, you are correct. Okay. Right, so if it increases from 1 to 2, so this is a boiling point curve. Because we have the liquid here, you have the solid there, and you have the gas here. So this is a boiling point curve, right? So if 2 atmospheres, it boils at 120, that's 120. And then at 1 atmosphere, it boils at 100. Okay, so the difference is 20. So that's correct. Correct. It increases by 20. It goes up. Okay, great. So far, so good. Page 228. 228. Number 58. It says the vaporization of solid dry ice to gaseous carbon dioxide by heating is an example of sublimation, A, B, evaporation, C, precipitation, and D, condensation. The vaporization of solid dry ice to gaseous carbon dioxide by heating. So you're changing dry ice to gaseous carbon dioxide.
A.
A, yes, sublimation, because you are changing from from solid directly to gas. So that's sublimation. A, correct. Sublimation. All right, so that is good. Let's see. Page 229. 229, number 33. Sorry, number 3. It's number 3. 229, number 3. What will be the likely result if all the bees and butterflies in an area were destroyed? Bees, butterflies in an area were destroyed. A, birds will have more food. B, more plant seeds will be produced. C, more flowers will bloom. D, fewer plant seeds will be produced.
A.
Look at it again. You are destroying all the bees, all the bees and butterflies in an area. What's going to happen?
I think it's D.
It's D. Yes, it's D. Fewer plant seeds will be produced. So if you look at this, remember bees and butterflies are the agents of pollination, right? So if you destroy them, then there'll be no pollination, or pollination will be reduced. If there's no pollination, there's no fertilization. If there's no fertilization, there is no seed production. So that's how you should think about this. So fewer plant seeds will be produced because the pollinating agents have been destroyed. Yeah. Right. Yeah. Let's see if another problem. All right, look at page 233. Page 233. Number 19 and 20. So you have 4 test tubes filled with water and placed in the test tube rack as shown in the diagram below. 3 of them are covered, each with different material. Then you have a thermometer is placed in each test tube, and the test tubes are placed in bright light for 15 minutes. In which test tube will the temperature rise most rapidly?
I would say A, the black paper.
The black, correct, correct. Yeah. So what was the reason? What reason do you have for that one? So
I
the
don't know, the
black one.
black always get the heat at the most, especially when it comes to clothes. So that's why I was thinking this way.
Yeah. So because the black one will absorb the heat, right? It absorbs heat when you put on black, something black. While the white reflects heat. White reflects the heat. So we're talking about here, that's where the radiation comes in. Like, you know, the aluminum foil, again, that one would reflect heat because like a shiny, right? And then no cover, yeah, no cover. So this one will reflect heat. So at least you know this out. It's going to reflect the heat, reflect the heat. No cover will absorb some heat, but then it's not going to absorb as much as the black. Black will absorb more. So that's why the black is correct. It's going to, black absorbs heat. So A is correct. So most rapid. All right, so number 20 is a diagram for a seed. Number 20 is a diagram for the seed. So it says that in the diagram that is given, which one is represent the stored food? So think of a diagram that we had for the seed. And you have this something like this here. And you say this is D. You're pointing to this as C. This as B. And then A. So which one would you consider to be the stored food for the growing embryo? Stored food. Think of the diagram that we about the seed. Remember the bulk is the stored food. Bulk of the seed is the stored food. So if you look here, A is going to store food. Stored food is A. The bulk of them. So it is A. Okay, let's look at another question. Let's see. Page 250. 250. Number 46. 215, number 46. So take a minute to look at that question. 215, number 46. All right, so if you got the answer, you can tell us. Anybody who gets the answer. Anybody wants to help us? You want to calculate the amount of heat. Okay, that's the calories, means amount of heat, right? So remember the formula, the quantity of heat is equal to the mass times the specific heat times the change in temperature, quantity of heat. Okay, the formula that we wrote down. Okay. So what's it? If you give the specific heat, that's 1 calorie per gram per Celsius. You know the mass, 30 grams. Then the change in temperature, the temperature is increasing from 25 to 75. So your change in temperature will be 75 minus 25, so which is 50. So that's a change in temperature. Okay, so with this information, you can find the quantity of heat. The mass is 30 times the heat capacity, which is 1, times the change in temperature. The change in temperature is 50. Okay. So if you multiply all this, then you will come up with 1,500 calories. Okay, 1,500. So the answer is C, 1,500 calories. Okay. Another question, page 251. Page 251, number 53. So take 1 minute to try 53.
C.
C. Okay, she's C. Look, let's try that and see. So in the pea plant, tallness is dominant over shortness. Okay. If 25% of one generation of pea plant is short, what are the probable genotypes of the parents? So you can work backwards. Yep, you can work backwards. So if you look at the first one, lowercase t, means they're all going to be short, so that's not good. If you try B, let's try B and C. If you try B, T, so we're going to have Punnett Square, so it's going to look like this. Okay, so we have something that looks like. So that's T, T, and then T, T.
Now I
So that
think it's
T,
necessary.
T, this with T, T, this with T, T, and T, T. So if you look at it, this is 50-50, right? 50%, 2 out of 5, sorry, 2 out of 4, this, and then this will be 2 out of 4 lowercase short. So it's like 50-50, which is not correct. So this is, so yeah, so B is not correct. So C, let's try C, as we said. So if we try C, you have T, T, and then we have uppercase and then lowercase. So this will be T, 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 T. This will be T, T, and then this will be what? So T, 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 this and that, and then this and that. Okay, right. So if you look on the outside, this is tall, tall, tall. No, no, am I right? No, no. This is, so they're all tall. So here they are all tall, right? 100% tall. So that doesn't match what they're saying, 75%, 25 short. So let's try D and see.
Is it D?
A D, right?
D.
Let's look at D. So D obviously should be the answer. So you want T, T, lowercase t, and uppercase and lowercase. So this is uppercase, uppercase. This will be uppercase, lowercase, and then T, T. Okay, right. So if I look at this, this will be tall, tall, and tall. So I have 3 out of 4 tall, and then 1 out of 4 short. So that's 25% short. So this matches the answer that I want. So the answer should be D. It should be D. So this is a question where I said you have to work backwards from the given answers. You do the crossing and see if you can match what they're asking you for. Yeah. Great. All right, let's look at 54. 54 doesn't match anything that we discussed, but let's look at that one. Let's look at an idea from the lab classes. Okay. Which of the following terms refers to the capacity of a microscope to distinguish between objects that are very close to each other? A, resolving power. B, staining. C, micro dissection. And D, magnifying power. So do some elimination for this question.
Is it A?
Is A correct? Resolving power. Or what we call resolution. Okay, resolution. So when I say resolution, it's the ability of the lens to distinguish between two objects that are so close to each other, right? So if I draw two dots, when they're so close together, if your eyes are not too good, you think they are like joined together, right? Even though they are two separate dots, you know. So some lenses are so good, they can tell you that these are two distinct dots. You know, if it's not too good, the result power is not too good, then it puts them together as if there's a single dot. Whilst in natural sense, they are two dots. So the ability of the lens to be able to tell you that you're dealing with two different dots is what we call the resolution, resolving power of that lens. You know, two distinct objects. Resolving power. The higher it is, the better. It gives you like a sharp image and distinction. Okay, good. Okay. So I think so far Let me see. There's another hit question. Let's see. And I will take 5 minutes break after this. Okay, let's look at page 297. 297. Number 44. It says a cross between a plant with red flowers and one bearing white flowers produces offspring, all of which have pink flowers. This is an example of A, incomplete dominance, B, Mendel's law of independent assortment, C, Mendel's law of segregation, and D, sex linkage. So which example is this? So red flowers, white flowers, producing offspring that have pink flowers. What do you think here? Anybody? Choose an answer.
Is it C? I'm guessing.
Okay, B. Okay, let's go back again. You have red flowers, white flowers, producing pink. Why do you think they are producing like a pink color instead of red or just pure white? Remember, we talked about the dominance, incomplete dominance.
Segregation.
It's incomplete dominance.
Incomplete.
Incomplete dominance. Remember, I gave that example under incomplete dominance. I use R and W, if you remember that part. So if you have R and W, then it becomes pink. None of them is more powerful than the other. So there's a co-dominance relation, a co-dominance there. So if you have, it's a mixture of the colors, pink color, under co-dominance. Okay, let me see. I have one more problem somewhere. Okay, go to page 341. Page 341. Number 38.
Excuse me, what page was that?
341.
Thank you.
Page 341, number 38. So it's a diagram, it's a phase diagram. Solid, liquid,
I think it's B.
and gas. Okay. So they have A, B here, C, D there, and then E, F here. All right. So you say which dashed line segment corresponds to the evaporation of a liquid? Evaporation of a liquid. So which one do you say? You say what?
CD.
CD, yeah, CD. Because this is a liquid and gas. Evaporation is changing from liquid to gas, right? So liquid to, so CD. That's CD, correct. CD. Evaporation. Okay, so that's how you use the phase diagram. Okay, good. All right, so one other question on page 343. Number 44. So page 343, number 44. So the following diagram shows 4 seal containers filled with water at various temperatures. In which of these containers is the vapor pressure the greatest? Vapor pressure the greatest. So this one is 10 degrees Celsius. This one is 30 degrees Celsius. And this one is 50 degrees Celsius. And that one is 80 degrees Celsius. So make a good guess for this one. Which one would the vapor pressure be greatest? Make a good guess. The vapor pressure depends on the temperature. It depends on the temperature. So vapor pressure depends on the temperature. Okay, that's a key. All right, so the higher the temperature, the higher the vapor pressure. Higher temperature, the higher the vapor pressure. Okay. All right, so lower temperature, lower vapor pressure. So here, the answer is D. Is D. So the one that is 80 degrees Celsius is the one that will have a high pressure. A, B, C, D. Higher. Right. And then let's look at the last question for this here. Page 344. Number 49. 344, number 49.
B.
Which one?
B.
Which answer? Is it B?
Yes.
Yeah, yes, B is correct. B is correct. Yes. How do you do that? So page.
Because it's only 100 milliliters and the temperature is 90 Celsius, so we need only like 10 Celsius to boil it, and we have less water, so it should be easiest. I mean, just, I don't know, logic.
Yeah. Okay. So yeah, basically, you're trying to calculate the amount of heat. It's a least amount, right? This amount of heat. All right. So let's just go to excuse me. Let's do the math and see. So the math, remember, we said quantity of heat is equal to the mass times the specific heat times the change in temperature. That's the idea you're using here. All right. So for the first one, let's call it 1. You are increasing the change in temperature to boil it. You have to bring it to 100 degrees, right? 100 degrees Celsius to get it to boil. So the change in temperature will be 10, as you said, all right, for the first one. Now the mass for the water will be the same as its volume, all right? The same as its volume, because if you think of mass of water, mass is equal to the density, volume times the density, right? Volume times density, that's what mass is. Okay, volume times density, that is mass. Okay. Now, the density of water is 1 gram per centimeter cube, is 1, right? So if I know the volume, let's say 100,000 times the density of water is 1. So this gives me 1,000. In other words, the volume is the same as, it's going to be the same as the mass of the water in this case, because it is 1. So we can use the volume as the mass. So the first one, the quantity of it will be the mass, which is 1,000, times specific capacity, which is 1. It's always going to be 1 for water. And then the change in temperature, here you have 90, so 100 minus 90. So if you do that, it's 1,000 times 10, and then you have 10,000. So that's how much calories you need for this one. Okay. Then the second one, 2. The mass will be 100 times the specific capacity, which is always 1 for water, and a change in temperature from 100 minus from 90 to 100, so that's also 10. So you have 10 times 100, so this will be 1,000, all right, for the second one. The third one, quantity of heat will be 1,000 times 1 times the change in temperature, which 100 to 95 minus 95, sorry. So that's 100,000 times 1 times 5. So this will be 5,000 calories. And then the last one, quantity of heat will be the mass, which is 100 times 1 times the change in temperature, 100 minus 50. So that will be 100 times 50, and that will be 5,000. Okay. So if you look at this, the smallest amount is B, 1,000. So that's why it's B. Okay. The least amount of heat that you need, 1,000. So remember, the volume for water is going to be the same as its mass, because the density is always 1 gram per centimeter cube. Okay, so the volume is the same. That's why you can do that here. And the specific capacity for water is always 1. Okay. Good. All right. So these are some of the questions that you should be thinking about for heat and the genetics type of problems, genetic problems. All right. Great. So I think we'll pause here, and then let's take 5 minutes break and come back and do some math. So let's come back. Let's. I'm almost 11:50. So let's come back 11:55, and then we'll do the math part.